Hi, everyone. I'm going to present the research that I've been doing over the last 4 years, and that has fallen under the broad umbrella of evaluating management strategies for coastal marshes from experiments to marsh-scale projects. And so the specific objectives were to increase understanding of and develop approaches to improve management outcomes of wetland ecosystems. And so in my first chapter, I evaluate salt marsh management techniques using existing literature. And in chapters 2, 3, and 4, I evaluate one specific technique using experimental and field survey work. And what that'll look like in presentation form is the first phase where we go over the need for management projects, which will talk about the marsh and their marsh's importance, as well as marsh loss. And then we'll close by thinking about the techniques that can be done to protect our coastal marshes in the face of these changes. Then in that second phase of the experiment, we'll dig into one kind of management technique called sediment addition, where we'll then address a number of different questions using my 3 chapters, chapter 2, 3, and 4, and we'll talk about that more when we get there. And so the ecosystem of our talk today is a salt marsh, which pretty broadly can be defined as a coastal wetland with salt-tolerant plant species that's affected by tidal hydrology. Here on the right, you can see this is a marsh out in Guilford, Connecticut, which in Connecticut, we have 2 tides a day, 2 high tides and 2 low tides. So water is constantly moving in and out of this marsh. And it's salty seawater, so the vegetation that exists there has to be pretty salt tolerant. So in this picture, we can see Spartina alterniflora, a salt tolerant plant. And we care about marshes because of the ecosystem services that they provide. Ecosystem services are the benefits that humans receive from nature, and they can be all sorts of different things. For example, they could be recreation. We use our salt marshes and coastal systems for kayaking and paddleboarding, things like ecotourism. We also value them for food and cultural importance, and storm surge protection, so they act as a buffer between the raging ocean and coastlines. And particularly important today is carbon storage. So all plants will take up carbon dioxide from the atmosphere and store it in their bodies and then in the ground through photosynthesis. And so they take it up through photosynthesis. And when they store it in the ground, they're effectively removing that carbon dioxide from the atmosphere. And that's important as the climate is changing. So climate change is occurring due to excess amount of anthropogenic or human activity that's causing releases of excess greenhouse gases into the atmosphere, such as carbon dioxide and methane, or carbon greenhouse gases. And so excess of these greenhouse gases is causing the changing climate, the warming of the globe, and sea level rise. So if we can remove any of that excess carbon, that can be particularly helpful. Also important for our conversation today is habitat. So habitat for a variety of different species and animals, and that is just incredibly important as we continue to move forward. And if we're talking about salt marsh habitat, we first have to talk about low marsh. So the low marsh habitat of a salt marsh is low elevation marsh dominated by the plant Spartina alterniflora. It gets wet with every single high tide that comes into the marsh and is dominated by this one particular plant that is salt tolerant and doesn't mind getting too wet. As you move up in elevation, you get the high marsh, and that's dominated by Spartina patens and Disticus baccata. And those 2 plants often will braid themselves together to look like this. Now, the high marsh will usually flood about twice a month, only during the highest of tides, and so it's not as wet or salty. And then you have the upland border, and that's higher in elevation, so it doesn't get wet nearly as much and is not as salty. And that's dominated by, in New England, Juncus girardii and Phragmites. So these are those upland species. We care about vegetation because it's used commonly as a metric for management outcomes. So when people go and do projects in their coastal marshes, they're doing them and using vegetation and how healthy or how much vegetation there is as their metric for how the outcome of the restoration activity was. So we know that our marshes are important. We kind of know a little bit more about them, but we are losing them. So here you can see the sea level has been rising. As well as coastal development. And that combination of coastal development and rising sea level means that you can see that the sea level is rising. You can see that the sea level is rising. Between the sea level rise and coastal development from humans, we have lost 35% of the world's coastal wetlands between 1970 and 2015, with even more being lost before that. And Long Island Sound, which is our location of interest today, is no stranger to that occurring. For example, we can see that in the 1880s, in red is where we had our estimated cover of salt marshes, and in green is where we had them in the 1970s. So far more red than green, which means we have lost our wetlands. If we zoom in on this particular area, Great Meadows Marsh in Stratford, Connecticut, we can see that this example, this green shown in the picture, is existing marsh that we can still see, and everything in red was lost to coastal development. These are Amazon and FedEx warehouses that can be found along the coast. And so that brings us to management techniques and how we can protect the existing coastal marshes that we still have. And the objective of my first chapter was to review evidence using studies on a wide range of coastal wetland restoration techniques to understand how effective various techniques are and where more research is needed. And so to do this, I leveraged the Conservation Evidence Database, which is a collection of expertly evaluated studies and actions that can be found on the internet. And so all of these actions are things that can be done to promote and protect aspects of the environment. They're evaluated by an expert panel reading papers to support the evidence of that action. And so that's kind of what this looks like. They'll read gray and white literature and score the effectiveness, certainty, and harm of the action based on the evidence in the papers. And my role in this was basically to take their database, which covers all sorts of different ecosystems, and only look for things that were salt marsh related. So I first filtered the database using their keyword system for marsh, swamp, conservation, and habitat of marine and coastal. I then reviewed the remaining list of actions and pulled out things that were specifically related to swamps and mangroves because we were only considering coastal marshes. And then I systematically went through and read the paper synopsis and the abstracts of each paper and recorded key information that was going to be useful later on. What we found is that there were 18 different actions that could be done to protect coastal marshes, and those were supported by 105 different studies. So the actions in this database include things under broad categories such as manipulating water levels. So facilitating tidal exchange could mean that they removed a culvert that was too small and created a channel to allow water to freely pass in and out of the marsh. Or they just installed a larger culvert, or maybe they controlled water through a sluice tide gate. The other possible general area of actions would have been to plant vegetation. So in this case, they are adding seeds or actually planting little seedlings and plugs, with the third kind of broad category being manipulating topography. So maybe they're adding a little bit of sediment or moving that sediment around and creating micro hummocks and so on. And so those are the three broad categories that each of these 18 techniques kind of fall into. What you can also see is on the left-hand side, they have been ranked as beneficial, likely to be beneficial, a trade-off, or having unknown effectiveness. So we have 2 techniques that have been ranked as beneficial with 12 as likely to be beneficial. And so basically the experts said that 2 of these actions definitely have enough evidence to benefit the marsh, to say that they benefit the marsh, but 12 of them don't have enough evidence. And in order to move from that likely to be beneficial category to being beneficial, what needs to happen is standardizing of evidence. And so person A may be measuring the outcome of their restoration through species richness, or the number of plants that are there, the kinds of plants. And person B might be measuring above ground biomass of plants. So they're both measuring metrics of plants, but they're not measuring the same thing. So the evidence is not enough to really support whether or not it is beneficial. So it's only likely. Then we also have a trade-off. So one of these techniques has a benefit to parts of the marsh and a detriment to others. And then 3 of our techniques have unknown effectiveness. So we don't know what they're going to do because only 1 or 2 people have studied them. And so those are areas where we need additional research. Overall, there are no actions that were found to be flat out harmful, which means anything you do as the marsh is either going to be neutral or positive. And so what we found is if we're using salt marsh species as our metric, they can be promoted by restoring marsh hydrology, adding or manipulating sediment, and planting vegetation, with again, 14 of those 18 actions having evidence indicating they could benefit the marsh. But we do need additional work, with the first being more information and more evidence to move things from that likely to be beneficial category to beneficial. So we need standardized evidence, so everyone needs to take the same kind of data in order for it to move from one to the other. And we also need longer term studies. So you can see on this plot that most of those studies happened on the lower end of the spectrum, with the most happening in 1 year and the median being 3. The shortest study was 9 weeks and the longest study was 30 years. So vegetation can grow pretty quickly. So you can definitely likely capture it in that 3-year span, but other parameters such as biogeochemistry are going to take decades to recover after that restoration activity. And so the trajectory of that management outcome is not going to be clear unless you're studying it for longer periods of time. And so that brings us to our takeaway today, which is scientific evidence indicates that actions that managers are already taking can promote salt marsh habitat. And of course, more research is always needed, with standardized protocols and longer-term studies to fully identify the impact of management actions on the marsh. And that brings us to the second phase of our presentation today, where we talk about sediment addition. So on the left-hand side here, you've got a diagram where the sea level is rising at the left, and you've got houses right on the coastline at the right. In between, you have a salt marsh kind of being squeezed like an accordion, as you have rising sea level on one side and permanent hardcore development on the other. And so the marsh is just drowning in between the two of them. We call this the salt marsh squeeze. On the right-hand side, we have sediment addition, where a layer of sediment has been added to the top of the marsh to elevate it out of the rising seawater. And that's really as simple as sediment addition needs to be. It just needs to protect and elevate the marsh out of the rising water. And so typically with these projects, dredge material is used. So our channels and marinas will fill with sediment over time because they're constantly transporting that in the water. And so as they fill, they become more shallow, which can make it challenging for boats to navigate through. So often these spaces are dredged in order to maintain good navigable channels. So basically people are going in and either scooping or sucking this sediment off of the bottom. And then it could be used for sediment addition projects, which is often what is done. And so when it goes to be applied, they can mix that sediment with water and spray broadcast it onto the marsh. As you can see on the left, that's one technique that's been done. But there are lots of different ways this can be done, and it also has lots of different names, such as thin layer placement, sediment slurry addition, and so on. The term I'll use today is sediment addition. And so despite being commonly used, this technique's actually been in use since the 1930s along the Gulf Coast, mostly in the United States. There are still a lot of gaps in our knowledge. And so that brings us to my second chapter, where the objective was to evaluate how sediment texture influences both plant biomass and pore water chemistry. And so what is texture and why do we care about it? Well, texture tells us the size of the particles of sediment. And in mineral soils, they're classified as sand, silt, and clay, where you can see a finer soil at the left and a gravelier soil at the right. And we care about what the sizes are because coarse grains such as sand will allow for faster drainage and decomposition and have more oxygen. But fine grains will have slower drainage, more nutrients and more organic matter, and less oxygen. And so kind of knowing what proportion of sand, silt, and clay you have can tell you how fast your soil is going to drain, how many nutrients it will have, and so on. And so in our experiment, this is what we were getting at. At 3 different sites along the Connecticut-Long Island Sound, we set this experiment up. At the westernmost site was Great Meadows Marsh in Stratford, Connecticut. Sluice Creek in Guilford was our mid site. And at the very east, we have Bride Brook at Rocky Neck State Park in East Lyme. Our experiment was organized in blocks to account for any spatial variability, with a total of 150 blocks, with 50 plots at each site. And we had 25 plots in the high marsh and 25 in the low marsh to account for those differences in vegetation and elevation. In each of those blocks, we tested 4 different textures and 1 control. Those textures were listed here in the table, with silt loam being very high in clay and silt and the least amount of sand. Then we have loam, which has more sand, sand, which has no clay and a little bit of silt, and then sandy gravel, which is a pretty even kind of mixture, including larger pieces of rock. In each of our plots, we dumped 10 5-gallon buckets of the right sediment mixture, the right sediment texture, and that brought each plot to an elevation of 7.5 centimeters of added elevation. And so with that, previous research has indicated that 7.5 centimeters of added elevation is what plants can grow through without too much trouble. So we're not dumping sediment so deep on the marsh that we need to plant vegetation because they're dying and crushed underneath, just enough that the plants are able to grow up and through. Our experiment lasted for 3 years, and we measured lots of different kinds of data, with the first being vegetation, specifically thinking about above ground biomass as our metric for management outcomes. Then we included pore water chemistry, such as sulfide, iron, and nutrients as our metric for redox conditions and nutrient availability. Soil samples, which more than what's listed here, but we have bulk density and below ground biomass as our metrics for the impact of addition to the physical structure of the marsh. And then we also counted crab burrows as our metric for changes in crab activity resulting from the addition, since crabs live on the marsh surface. Our experiment also collected greenhouse gases and dissolved greenhouse gases, but that's not included in my dissertation work. That's already been included in Dr. Maddie Meadow McDonald's work. She's now a Knaus Fellow in Washington, D.C. All of the analysis in this chapter was completed using a linear mixed effects model. In total, we took over 6,000 data points between 2021 and 2023. It's an immense data set. I unfortunately can only talk about vegetation, below ground biomass, and crab burrows today. And so when we went out to our plots, the first thing we would do is measure vegetation. We'd put down this PVC frame at the left and estimate percent cover of species. So how much of a particular thing was there. So 10% Spartina flora, 90% bare ground, or so on. Then in 3 random subplots, we would count and measure and identify stems of different species. And why would we do that? Let's watch a video.
Ready? Yes, ma'am. Alterniflora 5055. Alterniflora 5055. Alterniflora 5055.
So this is what the Salt Marsh team and I have been doing all week, identifying, measuring, and counting blades of grass. Why would we do something so tedious, you ask? Well, gathering this data allows us to understand the plant life in the marsh. From height, species, and density, we can calculate something called above ground biomass, which is basically how much plant life exists above the soil in a specific area. It can be an indicator of things like ecosystem productivity, or how big and robust the plants are, or the carbon storage potential. So we are practicing patience and resolve this week, counting individual blades of grass, all to gather important information that will help us understand the marsh better.
So we measured all of those different things to be put into an allometric equation, and that is just an equation that allows us to estimate the amount of above ground biomass in that plot without chopping all the plants out and weighing it. And so In March of 2021, this is what our plots look like. Pretty sandy, pretty empty. And you may be concerned that plants didn't actually grow through. But in August of 2022, you can see that they actually grew pretty robustly and rapidly through that added sediment, though there are a little bit of differences in between the treatments. And I do promise this is an actual plot. You can see we've got a little edge of the plot right there. So we did actually add sediment here. So as I said, there were some differences. In this figure, we'll explain those. In this first column, we have our first site, Great Meadows Marsh. Then we'll have Sluice Creek and Bride Brook. In the y-axis is above ground biomass, or the amount of plant life that's in the marsh above the surface of the ground. And along the x-axis is treatment. The top square is high marsh and the bottom square is the low marsh. Anything that's a star indicates that it was significantly different, so higher or lower than something else. And these were calculated with our linear mixed effects models, with a random effect for block and a fixed effect that is a site and treatment interaction, and then some post hoc tests. What I want you to notice is that silt loam in the high marsh at Great Meadows Marsh is significantly lower than a number of other plots out at the marsh. And so we had less above ground biomass in that plot. If we look at Sluice Creek, though, we actually see significantly lower above ground biomass in the sand plots, and that extends to the plots at Bride Brook. And that leaves us with two trends that I'll talk about today. The first being above ground biomass being significantly lower in the silt loam plots at Great Meadows Marsh, and in the sand plots in most cases. So first, we'll address the sand. Our sand texture had very little fine grains, with 0% clay and only 3.3% silt. Fine grains are important for nutrient retention, and plants need nutrients in order to grow. And so in order to continue to clarify this, we looked at our C to N analysis. So a C to N analysis tells us the percent by mass of the soil that is either C, carbon, or N, nitrogen. And nitrogen is one of those important nutrients. And so if we look at this table with our different amounts of nitrogen in them, we can see that our sand plots in both the high and the low marsh had the least amount of nitrogen compared to all of the other treatments. And so that indicates to us that yes, the sand did have less nutrients available to plants. That doesn't mean that the plants won't grow. It just means that having less is going to take them longer time to grow as much as the other treatments do. Now we can move on to the silt loam plots at Great Meadows Marsh. Great Meadows Marsh had an existing fiddler crab population, and silt loam has the most fine grains. Now, crab burrows usually occur in fine-grained sediment, and that's because they're pretty sticky and can keep the holes of the crabs open. So when we went in and we added these little squares of fine grains to the surface, we created the perfect habitat for fiddler crabs to exist in the marsh. And so their activity within each of those plots removed any existing vegetation that could have been there. And just to really make this point, you can see if now our y-axis is the number of crab burrows, in the silt loam plots at Great Meadows Marsh, we had almost 800 on average in our 1 by 1 meter plot, which is significantly higher than all of the other plots. So lots of crab burrows. What we can also say about this experiment is that, well, vegetation is not just above ground, it's also below ground. And so we measured that metric using below ground biomass. We took soil cores and looked at the roots inside. And now this is a similar figure, but not the same. Along the y-axis, we have the amount of below ground biomass, or the roots and so on. But now instead of high marsh on that top row, we have time, so 2021, and then along the bottom, we have 2023. What I want you to notice here is that in 2021, when we had just added the sediment, our control plots, or the native marsh that had plants in it, had significantly higher below ground biomass than the rest of the marsh, right? Because we had just added sediment to it, so there were no plants or roots there. But by 2023, even though we did see a growth of above ground biomass, we didn't see as many roots in those plots as we did in the controls. So the controls still had more roots, but not as different. But this is one of those things that we would need to measure over longer periods of time, right? So above ground biomass may grow pretty quickly within 3 years, like we saw in our first chapter, but it may take much longer for below ground biomass to kind of meet that reference level. And so the big takeaways of this chapter are that our findings suggest that effective sediment addition projects can be completed with a wide range of sediment textures as long as some fine grains are present. Also, findings suggest that if a site has a robust existing fiddler crab population, sediment addition with fine grain material may magnify the effect of crab activity on plant growth. And then unfortunately, I didn't get to talk about it for time constraints, but our pore water chemistry story is complicated. But generally, we see that fine grain treatments had more reduced parameters like ammonium and ferrous iron, but that wasn't consistent. And so that brings us to our third chapter, sediment addition and acid sulfate soils. Now, don't worry about what an acid sulfate soil is. I'm going to explain. It really simply is a soil with a pH less than 4.5 from the development of sulfuric acid. Now, these soils can form all over the world, but when they form in a sediment addition project, it happens likely because of dredge material. So sulfuric acid develops when dredge material, which is that sandy material that's been sitting on the bottom of channels that gets scooped up and dumped on the surface. When it sits on that bottom, it will accumulate reduced sulfide. And when it comes to the surface, that sulfide is introduced to oxygen, and the two will combine to create sulfuric acid. And so the goal of my third chapter was to identify amendments to prevent and neutralize the development of acid sulfate soils in sediment addition projects, specifically to measure how various amendments influence pore water pH, but also to quantify how other things can change, such as greenhouse gases or pore water chemistry. Now, these acid sulfate soils don't form in all sediment addition projects. Here you can see some examples where they have, and when they do, it's pretty devastating. So a pH of 4.5 is not an acid level that most plants will tolerate. For example, Spartina alterniflora, our low marsh species, typically likes a pH of about 6.5 in order to have the most robust above ground growth. So when we don't have a pretty consistent or neutral pH, it's too acidic for things to survive, and you just end up with a really moonscape kind of surface that looks like this. And so that begs the question of how do we prevent acid sulfate soils from developing? And you can think about that with how do you prevent when having an upset stomach when you've got too much acid? You take a piece of Tums. So we kind of took that Tums question and applied it to the marsh. The first way we thought about it is, well, we could amend the sediment by adding basically Tums, with pelletized lime being a form of Tums or calcium carbonate that has already been used in agricultural settings to neutralize low pH, and also calcium carbonate in the form of crushed shells. So these are two kind of Tums versions that we can use in the marsh to neutralize any acid. We also tested recycled concrete. It's not made of calcium carbonate, it's made of calcium oxide or quicklime, and that's just more efficient at increasing soil pH. And then our very last option was not to neutralize the acid, but actually to prevent the acid from forming. So mulch is made of organic matter that can buffer acid development, basically by taking up the extra hydrogen ions that are in solution. So it prevents the acid from even forming to start with. And so we studied these amendments through two phases of experiments. The first phase was in the lab, and it lasted for 13 weeks. We tested those 4 different amendments as well as 2 different controls. I manually added water in on a 12-hour inundation cycle, adding the water and then letting them drain just like the tides would. We measured pore water chemistry, such as pH, sulfide, iron, and nutrients every week, as long as a one-time greenhouse gas sampling for carbon dioxide and methane. Then we took our best performing amendment from the lab and into the field. And so we did this at Great Meadows Marsh for an experiment that lasted for 2 years. It was set up in a bucket style, so we had 10 buckets outside with our amendment and then our control. It received natural inundation because it was outside, and we sampled pH there monthly during the growing season, not during the winter. And we also yearly measured sulfide, iron, and nutrients. All the analysis in this chapter was done using a linear mixed effects model. And so in the lab experiment, the linear mixed effects model had a random effect for replicate and a fixed effect for date and amendment, asking the question, does pH change over time, and is it different between amendment? In this figure, if pH is the y-axis, with the bottom being the most acidic and the top being the most basic, we have an x-axis that is our amendment. And what you can see as I've been talking is probably that the pH in our recycled concrete is the highest pH compared to all of our other sediments. And so recycled concrete could be the most efficient at treating acid sulfate soils, and it moves on to our next round of testing in the field. So here, our analysis was similar with a model that used a random effect of replicate and a fixed effect for date and time, but now the model indicated that it was different, pH was different over both time and in between amendments. And so here now we have, again, pH along the y-axis, and along the x-axis we have time. In the solid line, we have dredge material, and in the dotted line, which is our recycled concrete. Our dredge material was our control. The recycled concrete decreased in pH through time, which makes sense because we are dissolving the calcium out of the concrete to treat the acids that's there. And so with every tide, that water that is going in and out is going to keep dissolving more, so we're going to decrease in pH. What you can also see is that the recycled concrete is significantly higher than the dredge material. So really just showing that it is really efficient at increasing pH. And that's kind of the big takeaway from this experiment, is that recycled concrete could be used to increase soil pH, and that could be useful for treating acid sulfate soils. And it's already being used and assessed to treat acid sulfate soils. So this is a picture of the restoration at Bride Brook in Rocky Neck State Park, which got completed over the winter. And so this is a site that had historically had issues with acid sulfate soils. And so they mixed in concrete at my suggestion to see if they could combat that development of acid sulfate soils. Now, we don't have the data of this yet. We don't know anything about what the pH is, but future work in my lab is going to address this with sampling over the summer. And that brings us to my last chapter, sediment addition in a marsh, where the objective was to evaluate ecosystem responses to the sediment addition by measuring changes in vegetation, soil, and greenhouse gases. So we did this by working with Connecticut Audubon and the U.S. Fish and Wildlife Service at a large-scale management sediment addition that they were doing at Great Meadows Marsh. And they were doing this because before the restoration, the area was a really nasty, degraded marsh. It was really sticky and muddy and full of water and rack, which is essentially just floated in trash and other dead material. So not doing its best ecosystem service self. So they added sediment to kind of elevate it out of that mucky area. They sourced their sediment from on-site, so it came from Great Meadows Marsh, along with some loam from a nearby garden center. At the deepest point, they added 5.5 feet, but then they also graded it at the edge, so you can see that here, to the marsh surface so that any plants that were there could grow up the slope. They also planted 3 different species, Spartina patens, Disticus spicata, which are our high marsh species, and then an upland species called Panicum virgatum. And really the goal there was to create high marsh of the future, taking this degraded area and trying to promote it into marsh. What we did in this experiment, it wasn't an experiment. What we did was to evaluate before and after the addition different metrics. And so we did 4 years of sampling. 1 year pre-addition at 13 different locations, which you can see in yellow, and then 3 years post-addition at 15 different locations. The addition occurred in this green box down here, and so we sampled at 10 sites that you can see in the green dots. on the addition, and then 5 sites in blue, which you can see off the addition. At each one of those dots, we measured percent cover of vegetation, and that was really to get at species composition. We also measured greenhouse gases such as carbon dioxide, methane, and N2O. And we took a bunch of different soil samples such as KCl extracts or soil nutrients and below ground biomass. We also measured pore water chemistry, sulfide, iron, and nutrients. And all of the analysis in this chapter was done with a linear mixed effects model. And so this looks like, this is a visual of the timeline. Before the addition, this is what most of the plots looked like. In 2021, they were pretty open, mucky, wet, bare ground. And then once they did the addition and added some plants, it looks better. We have more plant growth. And then through time, we see an increase in the amount of vegetation. And what that looks like in graph form is this, with percent cover, or the amount of ground that's covered by the thing, on the y-axis, and time along the x-axis. In this top row, we have bare ground, and in the bottom row, we have total vegetation. In the red line, we have the amount of either bare ground or total vegetation in the addition area, and then blue is the adjacent area. So if we care about the addition area, or where they added the sediment, we can see a decrease in the amount of bare ground over time and an overall increase in the amount of total vegetation, saying that if we added this sediment and planted vegetation, we can take a area that is primarily bare ground and turn it into vegetated marsh. And what that looks like for specific species is that we see a decrease in our low marsh species, which makes sense because we are elevating it. So it's not the same habitat where they can thrive. You can see a decrease in Spartina alterniflora through time, and that's our low marsh species. In terms of high marsh and upland species, we see an increase of those through time, which makes sense because we've created habitat that is high marsh and upland. We also see an increase in the amount of Phragmites. Phragmites is an invasive species that will often outcompete any native species, which is likely what we saw in this case. So Panicum virgatum is an upland species, just as Phragmites is, and it only existed in 2022, the year that it was planted. After that, we didn't see any more mention of it, any more existence of it. And so what we're thinking is that in the upland area, the Phragmites likely outshaded and outcompete the Panicum virgatum, as well as any other upland species, many other upland species. In terms of greenhouse gases, our models indicated to us that carbon dioxide was different both between area, but also through time, while methane and nitrous oxide were not. So I'll talk about carbon dioxide. You can see that we did not see an increase in greenhouse gas emissions after the addition occurred. It actually decreased and stayed low. We don't know if it decreased because of the addition, because the overall flux of greenhouse gases, carbon dioxide, decreased in both the addition area and the adjacent area. So we can't say for sure what caused that decrease. But we don't see any significant impacts, either time or area, on methane or nitrous oxide. And that leaves us with our big takeaways of this chapter. Our findings suggest that vegetation can rebound within 2 growing seasons with a significant increase in the number of species. And we also observed no increase in greenhouse gas emissions, and emissions remained low after 3 years, which is important when we consider greenhouse gases and climate change. And so if we step back and think about the key findings from all of my dissertation chapters, in the first chapter, we identified that many actions can promote salt marsh habitat, and that's supported by scientific evidence. In my second chapter, we found that sedimentation can be done with a wide range of textures as long as fine grains are present. In my third chapter, we identified recycled concrete as a potential amendment for acid sulfate soils. And then in my last chapter, we found that vegetation can recover quickly, and that adding sediment does not increase greenhouse gas emissions. With the broad implications of this work being that we have now deepened our understanding of management projects and sediment addition techniques, which is incredibly important as sea levels both in Long Island Sound and globally will rise, and that will affect the resilience and health of our marshes. And so The next steps is that the work continues. In my second chapter, the texture plots are going to continue to be monitored by Carly Shaw. And in my third chapter, the acid sulfate soils, as I had mentioned, is going to be taken on by future graduate students and our research technicians. I'd like to thank all of the people who were involved in the sampling of this process. It was an immense amount of data to collect in some really challenging conditions, very hot, muddy, buggy conditions, and I'd like to thank you all for being there. We couldn't have done it without you. And then, of course, my more personal thank yous. Thank you to my committee, and especially Ashley, who's been my advisor over the past 4 years. I'd like to thank the people of the NRA department, especially those who I've played D&D with and gone to the Renaissance Fair, my friends outside of the department, my partner Dave, my former roommate Alec, and then my family who stepped in on days where I did not have any field support. Thank you all very much for your support through this process. And thank you all for being here. I am happy to answer any questions if you have any. Thank you.